Okay, this is a great question. We got a 12-year-old girl with osteosarcoma, and her father has a brain tumor at age 30, and her aunt was diagnosed with breast cancer in her early 40s. Paternal grandfather also had adrenocortical carcinoma, and she's got this weird gene mutation on chromosome 17, and this is a mutation in a tumor suppressor gene. All right, so this is going to make us think of Li-Fraumeni syndrome, and the best way to test this is going to be single gene sequencing. So let's talk a little bit about these different diseases and what they're buzzy for and how we can test them. So A is going to be chromosomal microarray. This is best tested for copying the number of variations and microdeletions and microduplications. So we think here this could be like DiGeorge syndrome. So we'll scroll down so we can kind of see this stuff. Microdeletions like DiGeorge syndrome, which is a 22q11.2 deletion, like causing congenital heart defects, cleft palate, and hypocalcemia. Williams syndrome, which is a 7q11.23 deletion, that's like the elfin faces. Prader-Willi syndrome, causing obesity, hypotonia, hyperphagia, and Angelman syndrome, inappropriate laughter and seizures. So you can see those. However, Li-Fraumeni syndrome is just going to be a point mutation. The gene encodes for p53, and it's autosomal dominant cancer predisposition syndrome caused by mutations in the TP53 on chromosome 17p13.1, and it regulates the progression and induces apoptosis. So it's just going to be caused by point mutations in that gene. So that's why it's the answer. And so we talked about A, let's talk about B. So cytogenic analysis, this is going to be karyotype. So this is when you have large chromosome abnormalities like trisomies, translocations, aneuploidies. So think of Down syndrome, Turner syndrome, which is going to be 45, XO, Turner syndrome, they only have one. Klinefelter's XXY, tall male gynecomastia, hypogonadism. CML is another example of what you do for a cytogenic analysis when you look at the whole chromosome. And because CML has that 9 to 22 translocation, the Philadelphia chromosome. And fluorescence in situ hybridization, the FISH, identifies the known targeted chromosomal abnormalities, but does not detect the point mutations. So this could be also used for CML or cru de chat. So thinking about the fluorescence, you think of chronic myeloid leukemia, you think of the HER mutations, cru de chat, or DiGeorge as well. DNA methylation, that should make us think of Prader-Willi and Angelman. So Prader-Willi, P, so it's paternal imprinting defect, and then Angelman, M, so it's maternal imprinting defect. Happy puppet. And then the Beckwith-Weidman syndrome, 11p15 abnormal methylation that causes macrosomia and hemihyperplasia and a Williams tumor risk. So all of those would be methylation problems. So Prader, Angelman, Beckwith. And so the correct answer is the single gene sequencing, and Li-Fraumeni syndrome basically predisposes them autosomal dominant fashion to have cancer. So these other ones are useful for structural chromosomal abnormalities, but not for point mutations. So point mutations, think Marfan syndrome, FBN1 mutation, neurofibromatosis type 1, that's chromosome 17, Li-Fraumeni syndrome, TP53, that's also chromosome 17, cystic fibrosis, chromosome 7, that's CFTR mutation, and sickle cell, HBB mutation on chromosome 11. So the most important things, just to recap here, the answer choices scrolling up so you can see this. CMA, microdeletions, George, Williams, Prader. Karyotyping, that would be like big ones, Downs, Turner, Klinefelter, CML, FISH. That's fluorescence, so Philadelphia chromosome, cru de chat. Methylation, Praderman, Angelman, Beckwith, and sequencing would be Li-Fraumeni and Marfan's, neurofibromatosis, it's cystic fibrosis, and sickle cell. So hopefully this is helpful. If it was, like and subscribe.